Hi, everybody. It's really great to be here. First, I want to thank Mark Benioff and the Salesforce team for this opportunity to talk to you today about something I believe is one of the most exciting areas of biology and biomedical research, and that is the field of cancer immunotherapy. Today, we stand in awe of the things we've heard at this meeting, the way technology is transforming the world, the way artificial intelligence is going to change the way we analyze data. But I need to tell you that with all of these great breakthroughs, it's a distant second to the intelligence that our immune system has evolved over the many millennium. After a million years of evolution, after a tremendous amount of tinkering in the system, we now know that there is more data and more intelligence in the immune system than all of the computers that we have combined. Over the next 25 minutes, I'm going to talk to you about how do we try to harness the immune system to tackle one of the most devastating diseases of our time, cancer, and how do we use it to attack and destroy the cancers that we experience today. So right now, we all know your immune system's hard at work. Whether it's someone who sneezed next to you or coughed on the plane when you were flying in here, these microbiomes that sit around in the world, the viruses, the bacteria and parasites, need to be dealt with every day. And it's the immune system that is exceptionally good at eliminating and destroying it, and destroying every one of those bacteria and viruses and parasites. And this is a constant battle, a battle the immune system has been designed to exploit. Okay? It's very precise. It's capable of identifying and destroying foreign bacteria and viruses, even if there are single mutations that make the flu this year different from the flu last year. It makes one cold virus different than another cold virus. It's this incredible specificity and memory so that once you get rid of the virus or the bacteria, you'll never get it again, which is why we get vaccination only a few times in our lives. So once the immune system recognizes, it eliminates all of these bacteria and viruses. And now, for the first time, the research that's been done by our community using the immune system is now equipped to take on this deadly enemy of cancer. And what I want to tell you about is a story about how this occurred. So everybody knows someone who has cancer. 1 in every 3 women will get cancer in their lives. 1 of every 2 men will get cancer in their lives. 8 million people will die this year from cancer and every year following. That's how deadly this disease is. Okay. So about 150 years ago, people started trying to deal with this disease. At the beginning, we started cutting it out with surgery. Then we started poisoning it with chemotherapy. We bombarded it with subatomic particles through radiation. And we've hit it hard with targeted drugs to try to impede on their survival. People are living longer. Certainly, in some cases, people are being cured of the cancer. But these therapies ravage the body. They cause terrible side effects and horrible disease in and of themselves. And in fact, in most people, the cancers eventually come back and kill you. So why is cancer so tough to beat? In some ways, cancers look just like any other cell in the body. They start out just like a normal lung cell or a normal kidney cell or a normal skin cell. But then it starts mutating. In fact, cancer cells are a mutation machine. They start changing, they grow, they change shapes. They start migrating to different parts of the body. So a cancer cell in the skin can go to the heart, can go to the brain, can go to the liver, can go to the lung. And as they mutate, they escape all of these drugs that we try to give to destroy them. And just keeping up with it is impossible. And so cancer really turns out to be not just one disease, but many, many, if not millions of diseases, where each cell is its own cancer factory. And the difference between one patient and another is clear, but even within one patient, the cells are different from one another. So does this sound familiar? Doesn't it sound like the immune system's ability to detect and destroy viruses and bacteria and parasites? In some ways, cancers are just like that. So could we take the immune system and use it for good? So the first indication that the immune system could defend the body was some work done over 150 years ago by William Coley. He was a surgeon. He may not have been a great surgeon, or at least surgery wasn't great at that time because some of his patients got infections after the surgery. But what he found was that those patients that got infections often did much better than the ones that didn't get infections. And he hypothesized that maybe the excitement of the immune system in trying to eliminate the infections was involved. So he started infecting people on purpose. He started taking his patients and giving them bacteria after the surgery and looking to see what happened. And fully 10% of his patients, the cancers never came back, suggesting that maybe actually inciting the immune system was going to be a positive outcome. Unfortunately, most of Coley's peers at the time didn't believe it. They thought there weren't enough patients having an effect, and they really didn't think the immune system was doing much. But since the 1800s, we've learned a lot more. First of all, we've learned that the immune system is a highly regulated and balanced system. It's a system that has to deal with viruses and bacteria, and so we constantly are trying to do that, so we can't have too weak an immune system. If we do, then the viruses and bacteria will take over. But it's also a system that has to deal with its self-tissue, so that we don't recognize and destroy our self-tissue and get autoimmune diseases like multiple sclerosis and arthritis and type 1 diabetes and 80 other diseases. And if we have too much of an immune response, then we start getting those diseases. So at the end of the day, we don't want glitches in the immune system. We want to keep it highly regulated, highly balanced. We want to keep it in check through on and off switches. And in fact, the goal of the immune system is to keep this balance and to keep it functioning in a homeostatic way. Okay. So where do my lab and our work come into this? Well, about 2 decades ago, we started looking for some of these switches. We wanted to ask, what were the things that turn on the immune system when you want to, and potentially turn it off when you don't want it to? So we started out with turning on the immune system. And it turned out that each cell in the body, each T cell, which is the cell that's really important in recognizing viruses and bacteria, have a complicated set of on switches. They're like an ignition key followed by a gas pedal, where you press down on it and the immune system starts getting activated and starts going out, recognizing and destroying it. It's a very potent response, this on system for the immune system. In fact, one of the reasons that we reject organ transplants is because that system is so strong when you put in a foreign kidney or liver into somebody. So the first thing we did, and others, was to develop drugs that could block that on signal, that could prevent the immune system from going too strongly against in this case, organ transplant or autoimmune diseases like rheumatoid arthritis as an example. And that was very successful. But of course, the problem with that was it didn't deal with the other side of the equation. And the other side of the equation are the brakes, and trying to figure out what are the brakes that actually control the immune system as well. Because if we could understand the brakes, we could also unleash the immune system when we wanted it to. So I'll never forget the day that a graduate student of mine, Teresa Walunas, walked into my office to show me some data from an experiment. I think she had done it 4 or 5 times because she was afraid to come in because the results were opposite what we had expected. We thought we were looking at another on signal for the immune system, a molecule called CTLA-4. It's present on activated T cells, and it looks a lot like one of the other gas pedals. So we thought for sure that this molecule was going to be another on signal. But what she came in with data showing that if she blocked this on signal, putative on signal, it didn't shut down the immune response, it actually made it better. And it was really the first demonstration that these cells actually had brakes that could be engaged and shut down the response. That single result, that single day, changed my career for the rest of the time since 1994 when we made that discovery. And it led us to believe for the first time that maybe we could control the immune response in both directions, both the on switches and the off switches. It seemed logical that the off switches were important. Let's face it, I already told you that cancers look a lot like normal cells. So the immune system is designed not to see self as foreign. So these off switches made sense to be exploited by cancer. And in fact, it turns out that cancer does exploit these things by cloaking themselves with molecules that turn on those brakes and shut down the immune system. Well, less than 2 years after our discovery of CTLA-4 as the first inhibitor of immune responses, Jim Allison, working at the time at Berkeley, demonstrated that if you took a molecule, an antibody in this case, and blocked that brake, prevented the brake from getting turned on, not only did it goose up the immune systems, but it allowed the immune system to destroy cancers. And it changed the whole field because for the first time, CTLA-4 blockade, as well as a cousin molecule, PD-1, which had a similar effect, could be used as what we call checkpoint inhibitors, molecules that could turn off the brakes and allow the immune system to go when it otherwise wouldn't be going. Okay. There are now 4 approved drugs. The first approved drug was against CTLA-4 itself. It's a drug called ipilimumab. I know it sounds like a Dr. Seuss title, but all these things have weird names. Since then, there's 3 additional drugs that have been developed against the PD-1 pathway, and together these drugs have revolutionized cancer treatment, and now, for the first time, really showed that the immune system could be employed to get rid of cancer. Okay? And this is what it's done. This is what a graph used to look like for cancers, where we would develop new drugs, give them to patients, and they might live 6 months longer or a year longer. But inevitably, the cancer was going to take over. They'd mutate and change and go away. What these checkpoint inhibitors have done has really changed the outcome in many people. People who have melanoma, people who have non-small cell lung cancer, certain bladder cancers. And we've gone to a situation now when either used alone or in combination, we can see significant percentages of patients that have long-term, durable cures from these cancers, many out over 10 years now. If you take the example of metastatic melanoma, those people who had this disease had less than a 5% chance of living past 10 years. Now it looks like we're going to be in the range of 40% of those patients alive at 10 years. So this is a change. It's not just a change in the type of drug, but it's a change in the outcome of these kinds of drugs. One example is former President Jimmy Carter, who suffers from metastatic melanoma, which went to his brain. And he was treated with one of these checkpoint inhibitors. He recently said, "I had 2 weeks to live, and here I am building houses for Habitat for Humanity." This is a revolutionary treatment for those patients that respond. So it's very exciting. We now have a new set of armament in our toolbox to be able to go after and start to recognize, attack, and destroy cancers. And that would be great if that's where the story stops, but it doesn't. There's more. I want to tell you a story about a little girl named Emily. So in 2010, Emily was diagnosed with a cancer called acute lymphoblastic leukemia, ALL. It's a devastating cancer of the blood. In fact, after 2 rounds of chemotherapy and 16 months of treatment, one day her mom and dad, Carrie and Tom, came into the doctor's office, and the doctor told them that there was nothing more they could do, that there were cancers riddled throughout her body, pounds of tumor that could be identified in her blood. And they sent her home with not much left to do. But she was lucky. She got involved in a clinical trial at the University of Pennsylvania. It turns out that ALL, unlike the other cancers I've been telling you about, don't have as many mutations, so you can't use the checkpoint inhibitors to excite the immune system to go and attack them. So a colleague, Carl June, at the University of Pennsylvania at the Children's Hospital there, had an idea he'd been working on for 25 years. What if you could take the T cells out of Emily's body and use them to destroy her own cancer? I know it sounds like it's something out of a science fiction novel, but what if we took her cells and engineered a receptor in them that could recognize the ALL cancer cells? What if we could then take those cells that have been engineered and grow them up 1,000-fold and then inject them back in? What might happen? Well, Carl did that with Emily. They took her blood, they took her cells out, they put in this receptor that saw a protein on the cancer cell, they grew up those cells into large numbers, and they injected them back in. Now, I should tell you, Emily got incredibly sick. I mean, imagine what would happen if you put in all of these killer T cells into a patient that's got pounds of tumor. There was an immediate attack and response, and there was just toxic material spewed out of the cancer cells, spewed out of the T cells trying to kill the cancer cells, and Emily actually went into a coma, and they were worried that she was going to die. But a little while later, she came out of the coma, and the tumors were gone. And these so-called CAR T cells have now become an incredibly exciting area for research, where now patients are routinely with the ALL cancer and now other blood cancers are being enrolled in clinical trials that are designed to engineer their cells, put in a receptor, put them back in, attack and destroy those cancers. And in fact, something close to 70% of patients who are treated with this now are having their cancers go into remission, and we hope it'll last for a long time. And one of the reasons we think it might last for a long time is because these cells are like a living drug. They get into the patient, they do their thing, but they don't die and disappear. They just hang out. And if a cancer cell comes back, and if a cancer cell starts to grow, then those cells will be reborn, and they'll grow and they'll start attacking the cancer. So like in a vaccination, right? Like when you get poliovirus vaccine, if that poliovirus comes back, there are still T cells in your body ready to recognize that poliovirus and eliminate it. And that's what these cells do. Okay. So Emily's story is now in all the medical books. Here you have something that 6 years ago didn't even exist as a therapy, and now it's becoming a fundamental aspect of how we can think about treating cancers in the future. But as I've said already, and I want to say again, this is just the beginning. The new things that we're trying to do now are going to make this early stage of cancer immunotherapy seem primitive. For instance, one of the trials that's being done by the Parker Institute, which I'll tell you about in a second, is one where we try to combine the checkpoint inhibitors with these CAR T cells, but not to do it randomly, but to do it quite surgically and specifically. In fact, we're using this new exciting technology called CRISPR to go into the DNA of those cells that we've taken out of the body and selectively just eliminate the checkpoint inhibitor, so that the cells we put back into the patient have that chimeric receptor that'll see the tumor and destroy it, but it won't have the brakes anymore. So we won't have the ability to have the tumor cells shut down the immune system. So it's exciting. It's an area that I think will explode, and it's an area that's going to allow us to start treating multiple different cancers that right now can't get treated because these T cells won't make it into the site or get activated enough to destroy the cancer. So I want to end by telling you about how can we accelerate this? How can we actually make cancer immunotherapy a routine and reliable treatment for most cancers that we now get today? And this is where Sean Parker comes in. I know everybody knows Sean Parker, a very successful tech entrepreneur. But what people don't know is that Sean, at a very early age in his teenage years, suffering from asthma and other immunological-related diseases, actually started getting interested in the immune system, not just to understand his own disease, but when realized the power of the immune system. And when you get sick, you can tell how powerful the immune system is if you have asthma. How could you actually now tap into that to treat other diseases? And he landed on cancer. He decided right then that maybe we could use the immune system to cure cancer. When I first met him in 2010, he was already convinced that this was the way to go and that there would be ways to build an institute that would allow us to exploit all of the discoveries that were being made. Remember, in 2010, ipilimumab wasn't yet approved. We didn't have the PD-1 molecules out there yet. CAR T cells were just a thought in Carl June and other people's minds. So this was early days, okay? It seems like we've been doing it for a while, but it hasn't been that long. So in 2013, he began to work on the institute itself. And how would he form it? How would he develop a place where the best scientists could come, work together, and hopefully create new discoveries that could change the path of a patient's lives? Okay. That ends up now being the Parker Institute for Cancer Immunotherapy. Sean has challenged us to take chances, to make big bets, to take big risks. He's given us enough resources to try things differently than we've been doing before. We don't want to be constrained by the typical constraints that certain funding agencies like the NIH or industry might put, but be able to try things differently, to take people's wildest ideas and give a shot. Because it's when you do something you didn't expect. It's when you have a break when you thought you had a gas pedal. That's when big changes happen in science. And we've done this by trying to create a sandbox, a place where the people in it can share, trust each other, be confident that their work is going to make a difference. We've tried to create a sense of family, a core group of people that are comfortable working with each other, that are smart, that are committed and dedicated to advancing the cures for cancer. This is in the nonprofit space, so we're really focused on trying to move things as quickly as possibly into the clinic. What stays in Pisces, what happens in Pisces stays in Pisces. We want to make sure that the people around the table are really focused on the problem at hand. And what Sean says is he wants to hack cancer, just like he's hacked music and he's hacked other things in his life, and really come up with a better process to do this. So it starts out by having great institutions, and we have 6 of the greatest institutions in the country participating in this, from MD Anderson and Sloan Kettering to, of course, UCSF, Stanford, UCLA here on the West Coast, and University of Pennsylvania. These institutions combine incredible research and clinical assets and a history of translating discoveries into patient care. Each center has a director. Again, you'll see up here Carl June and Jim Allison, 2 of the people that I've talked about earlier. But each director at each of these centers are the top of their field, and they bring with them young people and innovative projects that otherwise might not be done. What we're trying to do is to facilitate bottom-up, bubbling up of new projects and new ideas, but yet do it in a team science roadmap that we've all created together to work on together. We encourage everybody to share ideas and papers and inventions and data as early as possible, well before they're published, so we can get a head start on what the next discovery might be. And of course, you can't do this in isolation. We need to partner with others. So we're partnering with different industries for new technologies and new innovative research that comes out of those industries. We're partnering with companies that have access to tools for computer and intelligent data analysis. We're partnering with other institutions that might have a unique perspective on how to identify and treat cancers using the immune system. And mostly, we're trying to make the Institute interdisciplinary. I already mentioned how we learned so much from autoimmunity infection that led us to think about new ways to treat cancer. Well, there are a lot of other scientific fields that matter. Nanoscience, metabolism, radiation biology, all of which impact on the ability of the immune system to recognize and destroy cancers. We want people who are involved in all of these fields. We want people that are involved in the microbiome. These are the bacteria that live in your blood and your gut and your skin, which we now know are essential to making these immunotherapies work. If you don't have the right microbiome, the checkpoint inhibitors don't work. What's the connection there? So we need microbiologists involved. We need geneticists involved. We need computer scientists involved. And the Institute is set up in a way that we can bring all of these people together to partner with us. And most importantly, we need the patients to be a partner as well. We need the patients to be involved in our science. We need them to be involved in our clinical trials. So why now? Why is this the right time for the Parker Institute? Why is this the right time to invest in cancer immunotherapy? Well, the science is there. I've tried to convince you we've made extraordinary advances there. But frankly, we're in an amazing revolution, biomedical revolution. It's not unlike the revolution of the late 1800s and early 1900s in the Industrial Revolution. We have more access to knowledge. We have more access to tools. We have more access to great ideas than ever before in biomedical science. The genomic revolution itself, the sequencing of the genome, so that every cancer cell can be sequenced and every normal cell can be sequenced, and we can see where the mutations are and what's different between the cancer cell so we can attack it with the immune system. We have computational technologies that are out there, created in fields quite far from medicine, which are technologies that can really be used to interrogate and change data into knowledge in our understanding of the immune system. And then we have leadership. We have people like Vice President Biden and his cancer immune shot, the National Cancer Institute, the FDA, who are all committed to new investments, and they're all committed to making new programs that can take the discoveries that are made in the laboratory and move them into patients. And finally, we have extraordinary commitments from the philanthropic community to support and accelerate the research. I've mentioned Sean Parker, of course, in the Parker Foundation, but people like Priscilla Chen and Mark Zuckerberg, and people like Mark Benioff and Lynn Benioff, who have spent so much of their time and energy trying to make science better, to make medical care better, to change the course of disease, that it's with the input of these people that I think we have a great chance of making a difference. So in the end, the immune system is truly an intelligent and very technology-based platform. The technology of the immune cell is enormously complex. But now we're starting to be able to decode that, and we're starting to be able to utilize it to attack cancer. Unlike the static brute force approaches we've used in the past of these therapeutic chemotherapies and radiation therapies and the like. Now we can start letting the body itself start dealing with the cancer cells. We're fighting fire with fire. We're trying to achieve durable remissions. And this is why I think we can succeed where others have failed. And this is what cures will look like. Thank you.
Ladies and gentlemen, please welcome Doctor of Philosophy, Associate Professor, Harvard University and Broad Institute, Howard Hughes Investigator, Dr. Pardis Salbetti.
Hi. Thank you very much for the opportunity to present today. I know there's so many amazing opportunities here at Dreamforce, and we really appreciate you taking the time to spend time with us. So I am very fortunate to be able to introduce members of a collaborative team that I've been able to join in the last few months. Who will be coming up to present in a moment are Susan Fisher and Arnold Kriegstein from UCSF. These are scientists who I have respected and admired for such a very long time, but that I never would have imagined that I would have an opportunity to work with. But there's something extraordinary about outbreaks or epidemics when they happen. We need absolutely everybody to participate. And and insights can come from anywhere. I don't think either of them knew that they would be viral hunters at some point, but they've become one. Because the Zika epidemic has been this extraordinary epidemic that has come to public consciousness that's now in the United States, with local transmissions in Florida and over 22,000 cases in Puerto Rico, with 6,000 to 10,000 women in Puerto Rico believing that they may have been infected, pregnant women. This is a massive epidemic that we need to be able to take control of. And the problem that often happens when epidemics occur is that it takes far too long to get everybody moving, right? As you see, it's been many months, almost a year into this becoming already, been signaled as an emergency, but yet Congress has now only released funds and tied it in strange ways to Planned Parenthood and other ways, making it very complicated to unlock. And that's where extraordinary benefactors like Mark and Lynn Benioff come into play. The reason why I became friends with Susan and Arnold is because of Mark and Lynn. They generously supported the efforts at UCSF and even on the East Coast at the Broad Institute where I work. They, brought these teams together, East and West, individuals who are experts in placenta and me who is a computational geneticist. All of this is possible with that kind of generous support. And it's so meaningful to me because it's so important. I'm deeply passionate about outbreak and epidemic response and about being able to get the expertise needed from everywhere. And you have the opportunity now to hear from some extraordinary folks about the work that they're doing on the front lines of the Zika epidemic. So thank you. I will sit here and listen myself.
Hello, everyone. I am going to start out by telling you how Zika spread throughout the world. It was discovered in 1947 in Uganda, and it was discovered by putting a sentinel monkey in the Zika forest. The monkey was to report infectious diseases, such as yellow fever, but instead the monkey got Zika infection. Thereafter, the virus spread throughout the African countries, and it ended up being the first human cases in Nigeria. After that, it spread throughout the East, and it ended up in Micronesia and Polynesia. As you know, it arrived in Brazil in 2014, and now it's spreading throughout the Americas, and it's arrived in the US. It's spread by the Aedes aegypti mosquito.
Susan, it's one over.
Pardon me?
So it's...
Oh,
yeah,
sorry.
sorry, just didn't want them to always have one slide behind.
Okay. It's spread by the Aedes aegypti mosquito. And this mosquito is really a very bad actor. It spreads all kinds of diseases. It spreads yellow fever and dengue. It's very easily identifiable by the white stripes on its legs and the fact that it's out during the day rather than at night. It's found throughout the tropical and subtropical parts of the world, but it's spreading due to climate change. No one thought that Zika infections would ever be a problem in the public health arena. This was because Zika infection produced no signs in most people who had it. And in people who did, the symptoms were minor, such as fever, malaise, and a rash. But this all changed very dramatically last fall, only last fall. And what happened was that a group of public health officials and doctors, OBGYNs in northeastern Brazil, realized that there was a cluster of babies born with microcephaly. In other words, heads that were too small. These babies all had one thing in common, and the thing they had in common was that the mothers had all been infected with Zika during pregnancy. By the beginning of only this year, the virus was isolated from the infected children. In the meantime, the World Health Organization declared Zika to be a global public health emergency. So, what is going on with Zika and pregnancy? Somehow Zika has learned how to cross the placenta. And my group has studied the human placenta for many, many years. It's one of the most understudied areas of all of human biology. Normally, it forms a protective cocoon around the baby. And as seen in the picture behind me, it forms a dense forest of branch structures around the baby. These dense structures are called chorionic villi. If we use an electron microscope to look very closely at chorionic villi, we can see that they're covered by another dense microforest of these branch structures. Their purpose is to regulate what gets to the baby. They let good things like food and oxygen pass to the baby, and they keep out bad things like viruses and bacteria. Zika is the first mosquito-borne virus to learn how to cross the placenta. This is truly a seminal moment in the relationship between viruses and humans. And we can actually see this happen in the laboratory. If we put placental branches in a dish and add Zika virus, we can see the virus, which appears here in green, infect the surface of the placenta. We think that this is the first step by which the virus plows its way through the placenta to reach the baby. Having this model in a dish enables us to screen for drugs that will prevent this transmission and the kind of birth defects that you will hear about next. Thank you.
My name is Dr. Arnold Kriegstein, and I've been privileged to work with Susan and also with Joseph DeRisi, an infectious disease expert at UCSF, to try to answer the questions about how the virus is actually getting into the fetus and what we might be able to do to protect the fetus from that virus. So in the next few minutes, I just want to briefly summarize some overview of what we've discovered and aspects of the virus that are really so unique. First of all, it produces, as all of you know, microcephaly in unborn babies, which literally means a small head. It's defined as 2 standard deviations or more smaller than it normally would be. But the reason the head is so much smaller is because the brain is actually smaller. The disease really doesn't affect the head, it affects the brain. And when we look at the brain of the babies with Zika, for example, shown on this slide, what we find is a really devastating picture. Not only is the brain smaller than it normally would be, and here on the left is a normal-sized brain, and on the right is a baby that was born with a microcephaly due to Zika virus. But the brain has really been devastated. The cortex, the gray matter of the brain is shrunken, is calcified, which is a sign of necrosis, of cell death. The ventricles, which are the spaces in the brain, which are normally very modest, are enlarged because the brain has gotten so much smaller. And these babies are devastated in terms of their clinical condition. They have seizures, they'll obviously have intellectual disabilities that are very severe, they'll have contractures. Sometimes the contractures are so severe that the limbs, when the babies are born, are unable to move because in the fetus, in the fetal environment, they weren't able to grow properly because the brain wasn't moving them properly. And in the pathological specimens that have so far been studied, you can see exactly what's going on to different cell types within the brain. So here on the left is a normal a brain of a fetus at the age of around 29 gestational weeks, and on the right is a similar baby brain that actually has now been infected with the virus. It's even older than the one on the left, but it's much less developed. It doesn't have folding that's normally characteristic of the human brain. There's a migration problem with the neurons, the nerve cells, when they migrate into the cortex. The calcifications of the brain is shrunken. And this is a really devastating form of microcephaly that I think initially wasn't appreciated and now is becoming clearly apparent that this is well beyond just microcephaly. And as a result, it's now referred to as a congenital Zika syndrome to sort of encapsulate the fact that it includes a lot more than just a smaller sized brain. So we at UCSF and others around the world have been trying to understand what goes on when the virus actually reaches this fetal environment, and what happens when those viral particles get to the brain, to the developing brain. And many labs have now used what are called cerebral organoids, an ability to take stem cells from adult patients and convert them into pluripotent stem cells and then make those into little self-organized nervous tissue that resembles or actually recapitulates some of the early stages of the fetal brain development. But this is happening in a dish, which you can then expose, in this case, to the Zika virus. And studies have shown that the Zika virus has some sort of tropism for these young neural stem cells. And as shown here, the green virus has infected the cells and started to kill them, which is the cells that are now expressing that red marker. And this results in a reduction in the size of these cerebral organoids, as shown here, a kind of reproduction of that microcephaly in a dish. And these preparations are now giving us an opportunity to study the mechanisms. And we at UCSF and others are now starting to look at the cellular consequences of what the virus is actually doing. And I just want to emphasize studies that were done studying how the virus affects the skin are giving us information that I think is translating to what happens in the brain. So the infected mosquito will bite you on the skin. The virus will spread and produce a rash by infecting neighboring skin cells. And it's been shown that the key receptor for that, and there are many receptors that can mediate viral entry, but the one that seems to be most important is called AXL, and that's been described in the skin. And so we asked whether this virus that uses that entry receptor in the skin might use a similar entry receptor to get to the brain. And for that, we actually turned to single-cell genomics. We looked at the profile of cells in the developing human brain and the genes that they express to see if AXL was on that list of genes. And this is a heat map. It shows genes that are expressed in these developing human stem cells and other cells in the developing human brain. And it turned out that AXL was very highly enriched in very specific subtypes of cells. And these were the neural stem cells astrocytes and blood vessels, not in the neurons, the nerve cells themselves, which was a little bit of a surprise. But the fact that AXL was highly enriched in the stem cells suggested that it could well be the way the virus was entering those cells and creating a smaller brain by destroying the progenitors that were actually making the nerve cells during early brain development. So we were able to test this idea by exposing cortical slices of developing tissue to the virus itself to see what happened. And what's shown here in green is a marker for the viral particles themselves, the viral proteins that they synthesize. And all those little dots in this image are cells that are heavily infected by this virus following just a 48-hour exposure. And the identity of these cells turned out to be, in this case, astrocytes. And one of them is shown in the middle of this image. In green are all these viral particles that are actually filling the cytoplasm of the cell and ultimately will probably destroy that cell. And it turned out that these are the cells that express AXL receptor. So we reasoned that if AXL was mediating entry of the virus, we might be able to block entry if we could block the receptor. And there are drugs available to do this and other methods to block AXL receptor, and we did that in our astrocyte cultures as shown here. And on the left is a control culture that's heavily infected now with these viral particles in green. And on the right, when we block the AXL receptor, we can significantly reduce the opportunity of this virus to actually infect the cells. So that strategy, by blocking the receptor to prevent the infection of the cells, could be a way of protecting the fetus. But the problem is that drugs available now to block AXL, which are being used, by the way, in cancer trials, have significant side effects. They can produce blindness, sterility in men. So they're not going to work as protective agents. So we turned with Joe DeRisi to a screen of already approved drugs that could be used in pregnancy, and just to see if any of them had an effect against the Zika virus. And we found at least one of them that we're very excited about, and that's maybe well known to some of you in the audience. It's azithromycin, which is a commonly used macrolide antibiotic. It turns out to be actually very effective in blocking the production of the viral particles and the death of cells. Shown in this assay, on the left are the infected particles that are untreated. They're all the cells that are expressing the green virus. Sorry. And with higher concentrations of azithromycin, as shown in the middle and the right-hand panels, you can nearly eliminate the infection and the reproduction of the virus in these cells. So this is a drug that's been used for 20 or 30 years. It's been used in pregnant women, and it's been shown to have no significant side effects. So because it's clinically approved and it seems to work in our assay, we've now, with the generosity of a donation from the Benioffs, initiated a clinical trial in Brazil to try to see if, in fact, this could be used as a way of protecting the fetus once the mother has been infected. Now, this is a really serious challenge, as hopefully you all appreciate. We're at the very early stages, and ultimately we're hoping that a vaccine will maybe eliminate the problem altogether. But this approach of trying to protect the fetus that's at risk is something that we think is an unmet clinical need that we're hoping to address. So thank you for your attention, but that's just a brief overview.
So I'm just going to take a few minutes and show a little bit about the burgeoning effort over on the East Coast to study this. We've come into it a little bit later in this collaboration and joined the team. My own work has actually not been in Zika and not been in the Americas. I've focused on work in Africa, where we were studying a virus called Lassa virus and began to recognize, even before the outbreak hit, that both Lassa fever and Lassa virus causing it and Ebola were circulating more widely than suspected. We call this question of emerging disease or emerging diagnosis. Are these diseases that we believe to be newly arising or rare in human populations and somehow springing up out of nowhere, are they truly emerging diseases, or have they been around for many, many years just going undetected? And as Susan mentioned, Zika was actually first discovered in 1947, actually one of those first viruses to be discovered in Uganda, and is likely circulating much longer, even well before that. And so one of the passions that my group has is, can we develop technologies that allow us to understand what things are circulating and detect emergent pathogens well before they become global pandemics? And can we then rapidly respond as they escalate to track them in real time? And things that I mentioned on a talk I gave on Tuesday is that now we had evidence that Ebola was mutating throughout the outbreak. And there was a lot of questions as to what those mutations happen. We now have new evidence that those mutations are making the virus more infective, more adapted to humans, and possibly increasing the way it was expanding. And these are frightening concepts, but at the same time, we recognize we have technologies to move that. And that becomes really important around the world, but of course in America we often think about what's happening in America. As I mentioned before, this is now this is a real U.S. problem. There are cases reported in almost every state. There are local infections in Florida, and Puerto Rico is being devastated by this disease. And so with the support from the Benioffs, we were able to quickly assemble. We don't work in the Americas, but when outbreaks happen, you get teams from all over coming together. And so we were able to quickly connect with a lot of individuals throughout the Americas who were working on this and who wanted to collaborate to have sequencing happen. And it was a real struggle to kind of get started. We only jumped into working on this because as months had passed and we were just continuing on our work on Lassa and Ebola, we recognized there wasn't much data coming out. The sequencing was not happening so fast. And so we started to work on it, and we put our entire team working on this for several months, and we could not generate sequence from the virus because it's at low titers in the blood when it happens, unlike Ebola where it makes a lot of copies of itself. It's at very low titers, but can do a lot of damage just with that. And also, it's often infected it for a very short period of time. So that makes diagnostics a challenge and understanding the virus a challenge. But we were very excited. Just a couple of weeks ago, we had a breakthrough. It was just kind of just in time for Dreamforce. We had this breakthrough, and we worked around the clock all night. I was a little tired during the talk because on that Tuesday when we presented, we were able to show that we now exactly went live with 33 full genomes of the Zika virus from multiple different countries. And this is so exciting, and again, cannot thank enough the generous support from the Benioffs to make that possible. And for us, it was very important to release that data in real time to the community. And these are just snapshots, because we at the Broad, we've really focused on releasing the data as soon as we get it, not after we've done a lot of our analysis and not after our publications. We're working on the back end on that now, but just making sure everybody can get it, because we need all, folks like Susan and Arnold to see it. If it becomes helpful to them, we need everyone to have access. But we can already see clusters forming. We can see that the virus indeed sort of is distantly related to a virus from Asia, and is likely, been there's a new expansion that's happened. This is a family tree that shows you how different viruses are related, but we can see a lot of interrelationship of the virus in the Americas and this sort of distant branch from up at the top from Asia. And we can also see how the different islands and the different countries connect to each other, and start to begin to see, we're digging into it now with a team from Florida, where we've an amazing team from Florida Gulf Coast University, where we have local infections as well as a lot of mosquito samples as well, to understand how these transmissions are occurring. And we can begin to investigate transmissions and lots of different, the mutations and what the mutations could be doing, and also importantly to be able to enable the types of diagnostics and vaccines that Arnold had mentioned, and as well as be informative to the therapies, and some therapies are based on the genome sequence. But this is incredibly important because there's so many pregnant women who are nervous now, right, and anxious in Florida, wondering, should I even get pregnant? And so what we need to be able to do, particularly if these kinds of treatments that Arnold is talking about come to fruition, is just be able to get diagnosis very early so that they can have all of the tools to protect themselves and to protect their baby. And so we've also, in the work that we've done, just the tech dev to get the sequencing, we have to develop assays to pick up what level of the virus is in the sample. And we've recognized that just by generating some sequence or using the sequence that's being generated publicly to develop new assays, we can rapidly develop assays that are much more sensitive than ones before. So that's another reason why this sequencing is so important, is to be able to rapidly detect it. And so already just what we have in the lab in our tech is better than the gold standard that's out there and available. And so to us, it's really exciting and important. We're very thrilled to have been able to finally crack this nut after many months, and to be able to do this with this extraordinary team. And with that, I just want to tell you, sort of give you the round of what we're doing, but I really want to finally kind of end by asking some questions, which I'm sure I have to be the one asking the questions, so I hope I ask questions that resonate with you, but I figured it's the kind of things that if you're coming to a group like this that you might want to know about Zika. So with that, we're going to start our panel. All right, thank you. Okay, so this is fun for me. My first question is to Susan. So Susan, you mentioned the fact that that this is the first mosquito-borne virus to cross the placenta. And that's obviously very frightening, and that image that you showed at the end, really, I mean, that's a profound image. Can you tell us a little bit more, because there are other microbes that have crossed the placenta, the sort of so-called torch viruses. Can you tell us a little bit more about the structure of the placenta and why certain microbes can cross and other ones can't?
Yes, so the human placenta might come as a surprise to many people, but the human placenta has a very unique structure. We think of using animal models, such as mice, to study things as diverse as the heart and the brain, because the mouse is a good model. The human placenta, the human head has required that we have a different structure to our placenta than other animals. And part of the placenta, I showed the branch-like structures. Part of those branch-like structures have evolved a cancer-like ability to invade the uterus and the blood vessels of the uterus. And in doing so, maternal blood is brought to the tree-like structures of the placenta. And with that blood comes the infection that can bring Zika to the baby. And other viruses do this as well. So a very critical and important worldwide problem is the ability of German measles to also travel to through the placenta to the baby's brain and cause abnormalities as well. So Zika is not unique. It's unique in how quickly it evolved the capacity to do this.
Great. I think we have time for maybe 2 more questions. So I'm going to ask one for Arnold and one for both of you. But Arnold, can I ask you, that what's really exciting is actually the fact that you can bring new technology to bear, and you use stem cell technology in the work you do, and it's so it's so fascinating why all types of basic science research are important, because it comes to be important at these times. Can you tell us just a little bit about that and how you brought that into bear?
Yes, it's an interesting convergence of technologies, as you mentioned, and stem cell technologies now allowed us to reproduce organs in a dish, and these are often referred to as organoids. And you can do this by taking, say, a skin cell or a blood cell from any of you in the audience, converting that in a laboratory into a pluripotent stem cell, a stem cell that really behaves like an embryonic stem cell. And then you can drive that down different lineages. You can turn it into a nerve cell precursor. And if you do that and take these precursor cells and allow them to self-aggregate, to actually start organizing among themselves, they will grow into what looks very much like a fetal brain. And if you start, of course, with human cells, this will be a human fetal brain. It lacks certain cell types. It's not a complete match for a young stage human brain in utero, but it does have many of the cell types that you'd expect, including the neural progenitor cells. And they look very similar, even in the single-cell genomic fingerprints that we do, to the cells you'd find in the normally developing baby. And so they allow us to study viruses like this in the laboratory in a dish in a way that we just couldn't do even 4 or 5 years ago. And this is a new technology, and so there are technologies that are still being improved to make it a better model of normal brain development. But for the first time in this disease, we've actually been able to show, I'm referring actually to we in the sort of a broad sense, scientists all over the world now, that this model system in the laboratory can actually predict and explain a little bit of what's going on in the normally developing human fetus. So the ability to do this with human cells instead of mouse cells is turning out to be extremely important. Mouse models of Zika virus are very different than the models in these human cell systems. In fact, in order to get Zika virus to get into the brain of a mouse, you have to actually use a mouse that has a very deficient immune system, because the normal mouse's immune system is so good that it gets rid of the virus anyway. So it's a very different system in a model system like a mouse than in the human system. And so this technology is really allowing us to study human cells in the laboratory in a way that we just couldn't do before.
Great, thank you. So it looks like we're actually out of time, and I think that I realize the audience is going to be left. I have so many more questions, actually, that we weren't able to ask, but we're all going to be here, and it just goes to show how much we still, there's so much that we still need to know. The work that they're doing is so important. Zika is still a mystery, but we can see the insights coming from the cutting edge. So I'm honored to be working with these guys, and I'm thankful to you all for taking the time to listen to our story. Thank you.